are a single cell.
Sorry, like in your lap. But no, you can just see him. It's okay.
The
Whatever.
chromosomal pattern is diploid.
You don't have to.
Half the DNA came from sperm and half from egg.
Yeah, she can hear it. She'll have my
Which
nose.
is accomplished there. And we cannot exist as a one-cell organism. So we must develop. That's 28. Development begins right here in the one-cell stage. And what you notice is zona pellucida is still there. You haven't even hashed out your little eggshell when you're one cell. What happens after this are these cleavages. Mitotic, cleavages, they're just cell divisions. You know what mitosis is, I think. We call them cleavages because with each cell division, I will draw them all. That's ZP. Zona pellucida, when you're one cell. Diploid. The first day, at some point, you'll divide into two cells. But you're stuck within the confines of ZP. The volume won't change. If you're going to divide one cell into two, you must reduce your size.
Bless you.
Call that the two-cell stage. And then it looks like a day later, call it day 3, you go from 2 to 4. Something like that. And the cell has to become smaller and smaller. And it's all, I'm writing 2N in the nucleus to remind you, this is not meiosis anymore. It's mitosis. We're done with that. Okay? So now you're just developing. So these are mitotic cleavages where you see a reduction in cell size. And it'll happen pretty rapidly over the next few days, and it doubles each time. 1, 2, 4, 8, 16, 32, 64. It's easier to nourish smaller cells, so it's advantageous. Okay. That's what's happening there. So by the third day, by day 3, you're a ball of cells, at least 16 cells, maybe more. You're a little ball of cells in ZP. I'm not going to try and draw it anymore. But day 3, and when I say day 3, this is, post-fertilization. Okay. I just put the name of the structure, morula, which is a term that means little mulberry. It's a little ball of cells, like the picture shows. Mulberry. Now, during this time, the isthmus of the oviduct is contracting. And it's keeping this conceptus, these structures, in the oviduct. We're not implanting yet. So assume fertilization happened at ovulation. Okay, day 14 of that cycle. Remember, you want to implant in the endometrium, you want to get that ready. You want to get well into the secretory phase before you implant. So you're kept in the oviduct and a little better. You don't want to implant too early. You won't take. Something won't take. So day 4, there are a little evolved cells, there's a little cavity on the inside that's starting to form. Those early blastocysts kind of float around still. Maybe it's allowed entry in the uterine cavity, but implantation hasn't happened yet. The day 4 is called the structural blastocyst. And the days are approximate. You'll see that there are differences when you look at a picture of development in your book and you compare it to a model. There might be several differences. You don't want to get hung up on that. You get the general idea of development. So day 4. Early blastocyst stage and I want to emphasize that it's free floating. Somewhere, maybe in the oviduct, maybe it's allowed in the uterine cavity. The point is, implantation hasn't happened. When you get to day 6, 7-ish, they say implanting blastocyst. Well, or just, this kind of gets to a more accurate picture of what we're seeing here. Fertilization happened here. When we went over the stages of fertilization, we'll review it. But then 2-cell, 4-cell, morula, okay, a solid ball, at least 16 cells in the morula, something like that. You're still floating around, and they say early blastocyst. This is when you see a hatching. The zona pellucida finally degenerates. Maybe there are secretions from blastocysts or the endometrial lining that help dissolve the ZP. I'm going to note that here. ZP goes away, zona pellucida degenerates. You can't have implantation if that's there anyway, so it's got to go away. And at around day 7, you start to have implantation. Okay, now, usually I like to go back and review fertilization, not go completely backwards, but I just want you to remember where we came from just yesterday. Okay. So this is backtracking a little bit, but I've noticed that reviewing fertilization now, before I continue with development, it avoids confusion on the test. Okay. So let's just answer them together. Take those if you want. Did this yesterday. Which steps are N plus N? When I say N plus N, there's more than one, which I would say. 2, 3. I think the best answer is 2 and 3. Yeah. 1, maybe, but it hasn't quite spit out the other half it doesn't need for the second qualifying. 2 and 3, I'll definitely say, you have discrete pronuclei, each with 23 single chromosomes. Which steps does zygote form? 4. When you have the fusion of those little pronuclei. Yes. What's the golden shell? Zona pellucida. It's not the cell membrane. Not the oval cell membrane. Very good. Like for this one, just look at it. The first parts of development. What's occurring in the day 1 cleavage? Mitotic or meiotic division?
Mitotic.
Get mitosis now. Now with meiosis, which cells, okay, I haven't talked about this one yet. Now, let's okay, so this one here, when we get to day 7, it implants. And I'm looking over the details of the blastocyst when we talk about how implantation occurs. And I put this slide here too, if you remember. That's the, I'm pointing out the day of ovulation, day 14 of the cycle, the menses cycle. And if we're implanting about 7 days after ovulation, assuming fertilization occurred at ovulation, what day of the cycle are we implanting? Okay, when did we fertilize? What day of this cycle did we fertilize? 14. So then what day does this implant? It should be 21 if you do math correctly. 7 plus, if you assume day 14, you should be on day 21. I'm right in the midst of the secretory phase. So that's okay. So when we say 7 days, that means post-fertilization. But when you actually implant, you assume it's around here. Obviously, you can't implant during menses, right? I mean, there's some contraceptive drugs that's how it works. Prevents implantation, blocks the progesterone receptor, whatever it is. So if there is a fertilized egg, it won't implant. You just lose it. Okay. Some contraceptive devices, they prevent fertilization, like a pill or contraceptive. It decreases the chance of ovulation, no fertilization. Some contraceptive devices allow fertilization but prevent implantation. Some we just call behavioral. Don't have sex. Or the rhythm method. You know, trying to figure out, okay, well, we can have sex right after my menses. You know, but we're talking about reproduction. We're not going to have a discussion about contraception. How does that happen? We hope it happens in, it's better if it happens up here, not down in the cervix. Implantation of a blastocyst involves you understanding, first, I already mentioned, this must degenerate. So that way you expose these outside cells. In the implanting blastocyst, take note of the trophoblast. Trophoblast is a term that means nourishment generator. This won't become the placenta, as we'll see. For now, just note this outer layer, the trophoblast cells, it's a prelude to placenta. Okay. The life-giving structure between the mother and the child, the baby. Now, the inner mast cells that they show, that is the embryo. That becomes the baby, the fetus, the embryo. I'll put it. That is you. The placenta is never a part of your body after you're born. They cut the cord and the rest of the cord shrivels off. So that's it. But the thing that sticks and implants is the trophoblast. So let's show a picture of that. Here's a picture of it sticking. The goal is to, this little thing has to eat its way into the endometrium. That's the goal. Here, it hasn't done that. It's just stuck. You see a little blob of glue right there. That's the inner mast cell. So this is kind of a small view. It's just showing you what it would look like to cut it all open. So this is a little blastocyst if you don't cut the ball open. It just sticks to it. Okay, they call it adhesion of the implanting blastocyst. So they say this is, again, about days 6, 7-ish after fertilization. So implanting involves a couple of steps. This step is adhesion. there's endocrine proteins on the surface that just stick like Velcro to the endometrial surface. First step is adhesion, that's the key word. I'll just note integrins as a surface protein that helps it stick like sticky Velcro. You just roll, stick, and then about a day later, you'll start to digest your way in. If you take a look on the inside of this adhesion stage, so what you see is that the trophoblast is the sticking part. The inner mast cells, it rotates inwards to match the surface, okay? So what I'm writing is inner mast cell rotates to the endometrial surface. It's on that side, flush against it. Just like the picture. Then you get differentiation about 8 days post-fertilization. Differentiation of the trophoblast. It differentiates into a syncytial trophoblast and a cytotrophoblast. Day 8. Day 8. It's still the trophoblast, just now the term syncytio, this prefix here, it's an acellular mass of cytoplasm. Basically, all the cells of the tissue fuse. There's no cell membrane structure, it's just a big blob of cytoplasm. A cellular mass of cytoplasm. There's no cell membrane structure, I just call it the giant blob. Some people call it just a fused tissue, right? All the cells, there's no discrete cell. It's just a giant blob. This is doing the digesting. It's digesting the endometrium. So it's trying to eat its way in. That's implantation. When you plant a little tree sapling, take it out of the pot, it's got some dirt on it, just lay it right on the ground surface, right? That's it, right? No, you dig a hole, right? And you shove it all the way in, right? That's what this is trying to do for itself. Digging a hole. Implanting itself. So there are things that this syncytial trophoblast is secreting, like tumor necrosis factor alpha. I told you something, it's making that endometrium necrose so it can eat its way in. This is the trophoblast that secretes HCG. HCG helps this process. Okay. You should be writing about HCG in your essays. Remember that? If you've completely forgotten HCG, ask a classmate, ask a friend. You gotta know that. Someone just shout out, what does it save? What does this maintain? It saves something.
Oh, the corpus luteum.
Yeah, it's the savior of the corpus luteum, so it doesn't die after 2 weeks. It's natural, so it enters the bloodstream. Let's talk about the cytotrophoblast. simply this trophoblast maintains the cellular structure inside of the cell. It's just a lot keeping the cellular membrane around the inner mass of cells. Maintains cellular pattern. You also see a specialization of the inner mass cell. It's not, there's some organization. Okay, there's 2 layers of cells. Again, that becomes the whole baby. Right now, it's just 2 layers of cells. That yellow layer and that blue layer right here. We call it embryonic disc. It's bilaminar. By how many layers? That's what bilaminar means. So note that for the stage. Day-ish. Bilaminar. Embryonic disc. Note epiblast, hypoblast. Also known as superficial or deep layer. So he's called that. So that is the baby. But notice that coming off of those stem cells, you get formation of amnion, these cells here, and these cells coming out here are gonna, well, they call it the exociliomic membrane. Simply put, it's something you don't stack. You start to see that here. I won't put it because you don't see it fully formed. I think that's enough to know. But let's say what the goal is here. This wants to preserve its own life. So one thing it's doing is it's secreting this gonadotropin to keep the corpus luteum alive. As long as that's alive, that'll keep secreting progesterone to maintain this whole environment. Otherwise, menses is gone. And the other thing is it needs its own blood supply here. So it needs to create spaces around it. It needs to make a blood vessel so it can be bathed in maternal blood. It needs a blood supply. It hasn't gotten it. Kind of like it's holding its breath. So I rotated the picture to make it look like the model we have in the room. I'm trying to read what it says here. I think this also says 7 days. Planting blastocysts. So it looks very consistent with the picture. Right? So identify that. If you look at this as a guide. Ooh, you're getting this out. Crap. That's a syncytial trophoblast. Makes sense. It's a giant block. It's a giant block. There's a giant block there. That giant block, that giant block. Okay. So then, what's this over here? What color is it? This is the the epi. This blue layer here, right? This is epiblast here. That's epiblast here. That's hypoblast here. Hypoblast here. For this one, I love tagging this. I don't know why. Every year I just tag this. Now, can you differentiate this from that? That's just the endometrium. Endometrium, this is the trophoblast. I mean, sometimes I just tell you I'm going to ask you this. You don't remember anyway. There's so much to know.
Was it lab, or did you ask us what day that is?
No, I don't ask that.
And another quick question, for the yellow ones, do you ask what days those are?
Yellow one?
The yellow ones over there on the shelf?
I give a study guide for that. But I usually don't ask the days. I think what I also have to study guide comes in class. Okay, this one, so we're day 9 and we're fully inside the endometrial wall here. And you see this is your trophoblast, trophoblast. You see the full formation of the amniotic cavity and the yolk sac. So I won't re-put everything we see each day, but I'll try to emphasize what's a little different. We're mostly implanted, okay? And, we start to see the formation of spaces around the syncytium. Syncytium is called the clue. The clue means, chamber, empty space. You want to fill those spaces with blood. What's that?
What happens?
Happens what?
What happens if there's no blood?
Well, then they'll die. You won't make it out of pregnancy. You have a miscarriage. Okay, well, let's see. We start to see amniotic cavity, yolk sac, femur. These cavities are not part of the embryo, so we call them extra embryo. Okay, extra, not part of the embryo. So, what we'll see is that the amnions will form a water-filled environment for the whole baby. It'll completely surround it. Okay. But right now, it's just a little tiny bubble on one side. It'll provide a water-filled environment. So understand that the baby's not breathing in the womb. That's why you can give birth underwater. It hasn't been breathing at all. You might think, doesn't it need to breathe? Well, it hasn't been breathing. And when it's born, it still doesn't know the concept of breathing. So if you give birth in a pool of water, that's fine, right? Then when you bring the baby to the surface, then it takes its first breath. So the water-filled environment doesn't have to breathe, doesn't have to use its respiratory system for gas exchange, because mom's providing the oxygen. The yolk sac that's basically for the genesis of blood vessels and blood cells. So again, the term I use to describe amnion yolk sac, if you ever see the term extra embryonic, it means it's not part of the baby proper. It's there though, and we need to understand it. I think that's all. There's nothing else that's really different here. I'm going to move on to day 9. And I want to kind of define, this is kind of the same time, different picture, about the same time frame on day 9, when you're mostly implanted. Now we'll teach you the 3 deciduous. Now, when you're pregnant like this, you no longer call the endometrium, you call it the deciduum. Basically, the decidua is the endometrium of pregnancy. like your deciduous teeth, you know what those are? Your teeth that fall out, your baby teeth. Decidua means that which falls off. Okay? Well, the whole endometrium falls off after you give birth. You give birth, and then you wait for the afterbirth. Basically, the endometrium. The placenta, but most everything else falls off too. There's a lot of blood loss. We usually call it decidua now. Now, this decidua, that's kind of underneath the embryo. That's the decidua physalis. Deciduas. Now, this one's really important. This is the maternal portion of the placenta. And it's always underneath the embryo. I mean, right now, the embryo is 2 layers of cells. You can visualize. We'll see other pictures. The decidua, that overlies everything on this side, decidua capsularis. This little cap. Decidua capsularis overlies embryo. The decidua parietalis is simply the rest of the endometrium. I put endometrium in quotes because technically we call it decidua now. It's just the rest of the lining. And it'll become more clear as we get a lot more development. And this embryo becomes fetus and it fills the uterine cavity and stretches it out. Right now it's literally a cyst. The size of a cyst. That's what they call a blastocyst. Day 12, you start to see the proliferation of the mesoderm. It is extra embryonic. It actually will help form part of the structure called the chorion. But for now, let's just kind of know extra embryonic. It's a proliferating mass of cells. So sometimes it's referred to as proliferating extra embryonic. And you start to see the lacuna nick blood vessels. The maternal sinusoids, that's a blood vessel. You start to see them. So I guess what we can note is the endometrial glands and the blood vessels, they start to fill the lacuna with blood and nutrients at about this time. So I'll say the lacuna begin to fill with nutrients and maternal blood. So gastrulation refers to that bilaminar embryonic disc. It's going to become actually a 3-layer stuff. Yeah, question, Daniel. About 3 or 4 months. Normally it's just 2 weeks. And corpus luteum of pregnancy, it's much larger than a regular corpus luteum of regular cycle. And after 3 or 4 months, you're hoping the placenta kicks off. And it'll secrete all kinds of hormones. All right, so gastrulation. That bilaminar embryonic disc becomes a trilaminar embryonic disc to form these 3 germ cell layers that form your entire gut, all your lean mass, brain, spine, everything. I just tried to distill it down to give you the impression these 3 layers become all your organ systems that are mature or in new form. So here's day 14, 15. For this gastrulation. So how I describe it is bilaminar embryonic disc becomes a trilaminar embryonic disc. That said, day 14-ish. How many layers do you see there? 2 or 3?
3.
I'm going to ask you this on the actual written exam. Not straight exam, but multiple choice. I'll just show you a picture like this. You see 2 layers of 3? 3? No, I see 2. Bilaminar, boom, boom. Okay, I know I wrote that. There we go. Well, I meant to show you this is still 2. But it will become 3, yes. Okay. So this is where we pick it up from. Day 14, 15-ish, this gastrulation will happen. So about day 14, 15 to day 16, this is gastrulation right here. Let's look at this picture. I'm trying to show you. Start with this picture here. I call it the 2 balloons. Okay. Top balloon is amnion. Yolk sac, but that bilaminar embryonic disc is those 2 cells, epiblast, hypoblast. Still bilaminar. And this is becoming trilaminar. So we see it starts to happen day 14, 15, and into 16. Where 2 layers becomes 3, basically. So let me describe to you how that happens. You get this primitive streak. The primitive streak will become the vertebral cavity. Right now, it's just a streak in the dorsal aspect. So what they've done is they've removed the 2 balloons. They've cut 2 balloons off. So you just get this disc. That will be the head. Then they cut the disc. And you're looking at this kind of sandwich arrangement of the disc with the primitive streak right down there. So that way you can see what happens here. The epiblast cells at around days 14, 15, cells will migrate into the primitive street and dive down into it, as shown. So this is describing gas relation. I'm just going to continue it up over here. Epiglottis cell migrate into the primitive street. And when they do, they kind of invade this bottom layer, hypoblast, and you turn it, they become the endoderm. Yeah. So think of this, the cell migration like the first wave, this first invasion. Cells become what will be known as the endoderm. Around a day later, about day 60, you get the second wave. And it's the same thing. Epiglottis cells migrate into the primitive street. And this cell layer actually becomes sandwiched between the layer above and the layer below. It's the one color red figure down there. I don't know if you can see it. That's the mesoderm. So that this second wave, it becomes mesoderm. And what you say is the cells that remain in the epiblast that haven't migrated, you now call them the ectoderm. Remaining epiblast cells are the ectoderm. And those are the three layers. The endoderm forms your digestive tract, the mucosa of the digestive tract, mesoderm. Now I'm going to move on to encapsulation. We're about 2.5 weeks in. I just have other pictures of it where a bilaminar disc is becoming a trilaminar disc, as shown here. As shown here, and trilaminar embryonic disc. And, another feature that I mentioned, all of the extra embryonic membranes that protect and nourish the developing embryo. And I've already mentioned 2 of them. These 2, if you have them in your notes, let's mention these 2. The allantois and the chorion. So this is day 16 after this day. The allantois, if you look at the left there, I think I'm pronouncing it right. This helps form the umbilical cord. It will stop. It will become the umbilical cord. I've also read, although I haven't seen a good animation of it, it becomes the urinary bladder. I don't have pictures of chorion yet, we don't have enough development, but let's just write it down now. This is the embryo portion of the placenta. So stick a pin in that, and we'll see it later. This is a good figure. In fact, I think this figure is on the lecture exam test. Multiple choice part. I got some questions about pictures like this. I don't know. Like I could ask you, which one shows the trilaminar disc, A, B, or C? C does. Can you all see that? Yes, it's something like that. Like, have you nicked a blood vessel here? 7.5, 12, 16. Can you identify that as a proliferating syncytiotropoblast? It's getting larger and larger. And on day 12, you start to see these lacuna form. When you nick a blood vessel, notice how the space is filled with blood. So we call them lacuna. Now they're called intervillous spaces containing maternal blood because basically the syncytium, it kind of like makes these finger-like projections. So it's like my fingers are the, well, the term is chorionic villus. Well, let me kind of draw what they trying to say. It's chorionic thing. You start to see it around 2.5 weeks. And the term is chorionic villus. And the spaces between them are, you can call them lacuna. Or intervillous space, either way. They're filled with maternal blood. This is the placenta. Okay, it's the embryo part. It forms the placenta. They don't put that. You just put it over here. If you can. Here I put chorion. Here I put chorionic villus. You know, these students don't get confused just because they changed the name, the phrasing of the word. It's still the same thing. It's describing the shape. So it's like the fingers that stick up, they're lined with I call it no master picture, sorry. But the black line I drew represents this proliferating syncytiotropoblast of the finger. Literally, that black line is but the green line I drew represents the proliferating extraembryonic mesoderm that's inside of it. What was the other kind of trophoblast? Not syncytial trophoblast, cytotrophoblast. That's still illustrated here. All the time, it's right here. It's well, sorry, it's in between these two cell layers. So I'm gonna draw a blue line. That represents syncytial trophoblast, which is on top of the cytotrophoblast. So this whole finger-like projection, that's the choriotic villus. Okay, that's the placenta. And the space in between is the lacuna. Lacuna without the E is singular. Of course, you want the lacuna to be filled with maternal blood in that space. Mom's blood. So we have a picture of that here. This is day 15 in our model. So it's somewhere between here and here, where it's still a bilaminar disc. Sorry, it's fuzzy. But even though it's fuzzy, identify that. It's maternal blood. Little villi here, there we go. The endometrium there, you can see there's arterial venous blood from mom's endometrium, the decidua of mom. Find that model in the room, and I just want to remind you that here is a picture of ovulation, gestation in weeks. You see elevated levels of HCG. For the first, it looks like it stays elevated to about week 16. Like I said, 3 or 4 months, so people just leaving alive. After that, the placenta, after a few weeks, the placenta should be taking over. You see the elevation of sex hormones. It really helps out with that. And that's really what I focus on. The placental formation. Most physiology books won't take development very far. You really need to take an embryology class. Okay? The physiology class will focus on maybe up to the development that we had, formation of the placenta, and maybe what happens to the mom with delivery. Kind of skipping the end here. Okay? So what we'll do is now we'll see the placenta come into full formation here. Pretty much we've got it right here. This is the placenta, where you have an interaction with mom's blood in a way to get it to the baby. Yeah. Blood is collecting in these spaces so it can get to baby. You just need to get some blood vessels up in these finger-like projections. Okay. That's all you need. Just blood vessels. Embryonic blood vessels. That way you can have an exchange of blood between mom and baby. That's all the sun is. Formed by the choriotic villi of the embryo, decidua facialis of the endometrium of mother. Okay. That is the placenta point right there now. You have it in your notes, just all spread out. Placenta means cake. Kind of looks, it's kind of pie shaped. We'll see, but right now the biology is choriotic villi. Decidua. Remember, there's 3 deciduas. Decidua facialis, the part of the endometrium that's underneath the embryo. Okay, because the part that's underneath the embryo is on the same side as the umbilical cord. Same side as umbilical cord. So we describe chorionic villi, we know what that looks like. So this is an interaction between mom's blood and baby blood. There's an exchange here. Mostly things like glucose and oxygen, and other blood gases across the placenta. So we see it forming here. Let's backtrack to day 13. When you're still a bilaminar disc, you see the two leaves. We have lacuna, they haven't fully filled with mom's blood yet. Okay. Here, 21 days, you see the embryonic blood vessels in the chorionic villi. Day 21. So if we want to put you drew it here, day 21, go ahead, I'm going to erase this. By day 21, you see embryonic blood vessels. Inside chorionic villi. Villi is plural, villus, US is singular. And we should see maternal blood inside the today filled with maternal blood. So day 21, you see the embryo take shape. You see the genesis of blood vessels in the yolk sac. There are some more things happening. I'm going to focus on just the placenta. Let's focus on that. Here are the placenta. You see the chorionic villus with the embryonic blood vessels, cytotrophoblast and cystiotrophoblast. Here you see the decidua. Okay, it's the endometrium of the mother. You start to see how if this big space is basically a lake of blood, you're going to have some exchange here. It's not a stagnant pool. This is blood circulation. So this lacuna filled with maternal blood is continuously renewed, right? Maternal blood is continuously renewed. I'll just make sure we noted all the things here. So we have chorion, cytotrophoblast, cystiotrophoblast, maternal blood. We've also noted an extra embryonic mesoderm with 8.2. Noted those three things. I'm just reminding you that HCT is going to maintain that structure for a few months. So here is 4.5 weeks. Move along here. Here we see more development of the baby. And I'm really going to focus on how all the extra embryonic things are helping it. You can see the amnion is fully providing a buoyant environment for the baby's yolk see the umbilical stalk. Okay. You see all the chorionic villi going all the way around. This becomes the umbilical cord, and that's really all you need. You still have complete nourishment, completely by the baby. There's all these arteries, internal arteries, spinal arteries that are freshly supplying the blood. They don't really, well, they do show a vein. They don't show a mom's vein here, but there's venous blood that's taking it away so that these lakes of blood continuously renewed. So we have our chorionic villi going all the way around. This is a full functioning placenta at 4.5 weeks. We have a model of this class. What is this? Okay, you call the amnion, or it's also known as the amniotic cavity. Don't let that variation. This is the amnion. Amnion just means the whole space for the baby. Sometimes they call it the amniotic cavity because it is a cavity. Two would be the umbilical stalk. So what's this thing?
Yolk.
That's the yolk. What if I put a little pin here? I just said identify. What would you call it? Yeah, chorionic villus is right. That's what I would be going for. Some students would think, oh, he's pointing those blood vessels. Blood vessels. Those are blood vessels, but who do they belong to? Mom or baby?
The baby.
Embryonic blood vessels. So if you just put blood vessels, I'm not going to give you full credit. You should know if it's mom or baby. But I would accept that you know that they're embryonic blood vessels. But really, it's the chorionic villus has to be instructed that this is illustrating. What if I just put this here and said identify?
Lacuna.
Maternal blood. Okay. Or you could say lacuna with maternal blood. In the model, they do show a good job with mom's oxygen-rich blood, but can you see how it becomes oxygen-poor? Okay, because there is gas exchange happening in the villus, and then maternal blood will be swept away here in the vein. So those lakes of blood are continuously renewed. So very well. What if I just put a sticker on this? What would you call it? It's the visual basalis. It's on the same. It's underneath the baby. It's on the same side as the umbilical cord. Where's this decidual? Capsularis. I think that's all I see. This is a 13-week fetus. Notice how the chorionic villi on the capsularis side, they've completely gone. We've completely flattened out the chorionic villi on this side. They're not there anymore. You see the full formation of the umbilical cord. We see the term is chorion leve. Means flat. Basically, it's this here. Okay. You see how on this side, the chorionic villi get flattened out? When they're completely flat and gone, that's what you call it. Okay, because this term means level, flat. There's no villi on that side, the decidua capsularis side. So there's chorionic villi with the decidua capsularis. That along with the amnion, you also see those three layers. Is what you see on the side opposite the state. No more chorionic villi. Now, when you reach full term, you could artificially rupture when we say the water breaks. Well, it's not just the amnion; it's actually those three layers, and you can help induce labor. The physician can by poking these, rupturing these, artificial rupture of membranes. I mean, this is when you're full term and you want to induce labor. The OB could use your fingers. There's tools that could do it. And just kind of poke inside the cervix and try to rupture them. It's very painful because it induces uterine contractions when you do that. So when we come back from break, we'll talk about the physiology of the
Wasn't that bad?
Concepts are fine. There's just a ton of new words. It is. This is a foreign language. Why did I have to take